Hello, and welcome to your virtual classroom lecture on Chapter 6, the Endocrine System. First, I'd like to review with you the learning goals for this chapter, which includes the endocrine system. Major endocrine organs, their structure, location, and function, including the pituitary gland, adrenals, thyroid, and parathyroid glands. Chemical classification of hormones and hormone actions and interactions with their targets. I will try to summarize these major learning goals with you in this short video recording. One key feature is the use of the circulatory system for delivery of these essential chemical ligands to every tissue in the body. Here in figure 1.3, we see blood flow to the gland that is going to be secreting a chemical ligand and using the bloodstream to access its target tissue, which may be some distance away. The Homework 6 assignment, already posted in Canvas, will help you to associate the various endocrine glands with their chemical ligand and its function. Let's review the way a chemical ligand can interact with its target cells or tissue. First, we need to decide if the chemical ligand is water-soluble or fat-soluble. Water-soluble hormones need to have the receptor facing the ECF compartment. So the protein receptor is embedded in the phospholipid bilayer, and its active site is facing the external interstitial fluid. That way, when the ligand is delivered by the bloodstream, the ECF compartment will be filled with this chemical ligand, and the ligand will bind to the receptor outside of the cell. If you have a fat-soluble ligand, like a steroid hormone, thyroxine from the thyroid will also behave this way. These are fat-soluble. They don't like to be in water, so they are going to be able to diffuse directly across the plasma membrane using those fatty acid chains and gain direct access to the inside of the cell. So fat-soluble hormones will not have an external receptor. Their receptor will be found inside the cell. I placed a big red circle around the steroid hormone or fat-soluble type of chemical ligand interaction with the cell. It diffuses into the cell and finds its receptor in the ICF compartment or maybe even in the nucleus. So the nuclear membrane has the same phospholipid bilayer. It is likely that a hormone that has fat-soluble characteristics could access the nucleus and affect things like mitotic cell division. These hormones can cause a lot of cellular changes with these different target cells. I mentioned stimulating mitosis or cellular division, but they're also able to alter plasma membrane permeability, stimulate protein synthesis, activate or deactivate enzyme systems, and also cause the cell to start secreting other products. So these hormones have a huge effect on their target tissues at the cellular level. We've already studied how the nervous system uses chemical ligands to communicate from neuron to neuron across the synapse. The endocrine system does the same thing, but it has to use the circulatory system to deliver its chemical messages over great distances. One of the differences between the neurotransmitters and hormones is not only the bloodstream being used, but we also have to have a specific receptor available for these chemical ligands once the ligand arrives at the target. And there must also be a method of deactivating the signal. Hormones are tightly regulated, so don't forget about negative feedback mechanisms. Target cell activation depends on three major factors. One, the rate of secretion, or how high the blood concentration levels are going to be. The relative number of receptors found on the target cell. So more receptor sites means you're more likely to have the signal bind and carry out its action on the cell. If you have fewer receptors, you're less likely to have the signal bind and carry out its message. And also the affinity for those receptors to the hormone. Affinity is that attraction between the receptor and the chemical ligand. How likely are they to find each other and bind, usually through hydrogen-hydrogen interaction or van der Waals. The term upregulation is going to mean the target cell is going to form more receptors in response to the hormone. The opposite is true during periods of downregulation. The cell can lose some of these receptors. So the cells or the tissues can become more or less receptive to these chemical signals. From homework 6, you've probably already reviewed most of these glands that you see in the diagram here. We've got secondary endocrine glands and we've got primary endocrine glands, and your job right now is really to become familiar with the hormones listed and understand the difference between the different hormone functions. So it's really up to you right now to not only do this homework, but also maybe start a list of each gland on the left and maybe its function on the right, and keep that as part of your notes as if you were in class doing this work with me. And you can also put next to target tissue is hormonal response. So you could actually do a 3-column note sheet to keep with you and build your study guide. One of the things I want to discuss quickly is dual function. Many of these endocrine glands serve dual purpose. They'll secrete more than one hormone. And you'll need to make a note of that as well as you're forming your list and doing your studies. One example I can pick on, and there are many, is the pancreas. The pancreas secretes digestive enzymes into the duodenum at the base of the stomach during a meal. Those digestive enzymes help you digest your food. But more often we think of the pancreas as a secretor of insulin. So insulin's going to flood the bloodstream during a meal. And that's going to help the cells absorb glucose. The other hormone the pancreas secretes is glucagon. Glucagon is only around when you're in the unfed state or starvation mode. So try to remember, glucagon is only around when glucose is gone. I hope this helps you get a handle on Chapter 6 and get the most out of your current homework assignment. More to follow.